going to get a triple paper shout out tonight. A Morph-Electro-Transcriptome. I'm going to get a triple paper shout out. Good evening, Pittsburgh and universe. This is JC on a bike. My name is JC and this is Journal Club on a Bike. I'm going to get a triple paper shout out tonight because there are 3 large data sets that were recently released this week or last that try to dissect cell types in the cortex. And of course this has been done lots of times. Probably started with Samoji in the hippocampus with interneurons, and then Henry Markram's lab did a couple things with the E code in cortex, but in babies. And then now we have 3 papers this week sampling excitatory or inhibitory or both, and trying to combine morphological, which is the anatomy, the physical shape and structure of the cell, with transcriptomic analysis, so what genes are being expressed, and the electrophysiological subtype. And so if you call this a Morph-Electro-Transcriptomic, then there are 3 papers that are attempting to put the classification of cortical cells into the context of a Morph-Electro-Transcriptome. And so the first paper is by the Hanson lab at Genentech. They're in California. And what they've done is fact sort cell types. And so fact sorting allows you to pull fluorescent cells out of a sample using a machine that can sort the fluorescent from the non-fluorescent cells. There may be one issue with this is that when you digest the tissue, in order to create the cells that you fact sort, there's quite a bit of physical and mechanical things that occur during that process, and I'm not going to claim to have read the paper in detail enough to know what extent they have tried to mitigate those effects. But if you digest a slice into dissociated cells, there's no question that in my mind anyway, there's no question that the genetic complement being expressed will inevitably change. So this paper does fact sorting and then does RNA-seq on the cells. And RNA-seq also requires you to pool cells together, so I presume that a lot of times for these samples they are pooling numbers of cells into the group and then doing RNA-seq on that group of cells. It's not single cell RNA-seq, I don't think, although it very easily could be. If they were doing single cell seq, RNA-seq, I don't think they would use FACS then, so I'm guessing it's not. And then they're going to look at electrophysiological and morphological subtypes that coincide with these transcriptomic types. And so in some ways, maybe the way that they did it works, right? They first determine the transcriptomic type, and then they do the morphology and the electrophysiology and slices. And so in a way, they get single cell data tied to groups that they already identified. So in a way, it's kind of a cool way of doing it. It's very different than the single cell patch seq that's done by the next 2 papers, Tolias' lab and here we go. So you're going to either cut me off or you're not going to cut me off, but you can't do both, because that's crap. So the second paper I want to mention is from Andreas Tolias' lab, where he does it in M1. He's out of Texas. And so what these guys are going to do is look at phenotypic variation between and across transcriptomic cell types in the motor cortex in particular. So the Genentech paper, Gentech paper, the Hanson lab paper is really doing, what are you looking at? I have the right of way. You're the one who's running the red light by going left in front of everyone. You can call it a Pittsburgh left, but it's running a red light. So the Andreas Tolias paper does patch-seq. So that's really patch clamp on an individual cell in a slice, pull that soma out, and sequence the DNA. Now, the details of patch-seq, depending on how you do it, vary a lot. So you're going to need to dig into the details of these papers. That's not what this ride is about. This ride is to give a shout out to these papers to make sure that nobody misses them. So the first one again is fact sorting. So the transcriptomic cell types are determined by groups of cells pooled out of different genetic backgrounds. And this one is single cells. And so they get a lot more data that is single cell oriented. So they get individual morphologies, individual electrophysiologies, and then individual patch-seq data that they then cluster. So there's a lot more variability in this paper and a lot more variability to discuss, but it's motor cortex and it's patch-seq in combination with morphology and electrophysiology. So I think it's a step above the Hanson paper just for that reason. And the final paper is from the Allen Brain Institute. It's a monster paper. Of course, it's a monster paper. They've got a lot of people. It's probably got 3 times the number of authors that Andreas Tolias' lab had on his paper. And certainly many more authors than on the Hanson lab paper. But to patch individual neurons, fill them and reconstruct them, and also do the transcriptomics, well, you're going to need an army. And so that's why the Allen Brain Institute paper is so nice. And so there's 2 patch-seq papers and one fact sorting paper. But they all are trying to do the same thing. They're trying to give us some understanding of where we are in terms of cell types and whether that's a viable option, types. And more labs, I think, are coming to agree on the idea that it's a manifold of cells, and it's a spectrum. And so there are branches to the tree, but there's a lot of variability as you approach the end of the branch. What are you doing? Are you really going to go slower than me because you think your traverse is so wide? It's not, just so you know. Oh God, give me a break. I can't stand it anymore. Such an idiot. Crap driver. All of these things are available as a web resource. You're an idiot. And so you can download these and explore these data sets and see how well they compare to each other. And I suspect people are already doing that within the labs. to try and get an idea of whether or not we're moving toward a common goal and whether or not we're moving toward a common consensus of whether or not we need to classify anymore or not. Because one of the things that really bothers me about this, and I think you can look into Cliff Kentros's paper and some other papers if you look up enhancers, is that although they know, and the Allen Brain Institute has even started doing it, they know that they can break down cell types in the cortex and in the brain in general using enhancers in a much more specific way than they can with promoters. Come on, dude. You need to frickin' drive. And so, in other words, what I mean is that if they try to pull IT or PT cells, for example, in layer 5, and they find a promoter that's specific for those cells, as they have done, what they find is that they label those cells in all brain regions. Well, what Cliff Kentros's work and all the work done in developmental biology has shown that actually it's the enhancer complement that is changing to differentiate between these cells in the adult animal. Yada, yada, yada. I mean, look at these people drive. She's carrying a coffee in one hand and sleeping on the chair in the other. So thanks for joining me on my ride. Those were 3 papers tonight. Just a brief introduction / shout out to the Hanson lab at Genentech that did a fact sorting / morphology and electrophysiology characterization of cells in the cortex. Andrea Tolias' lab did motor cortex with patch-seq morphology and electrophysiology, and then the Allen Brain Institute released a pretty juggernaut of a paper of a very similar patch-seq, but in visual cortex. So motor cortex and visual cortex have kind of been done by laboratories that are using the same techniques, and so I think those are comparable data sets more so than the fact-sorted one, the first paper. But the 3 of them together are trying to classify neurons, and I think the 3 of them together are starting to show us that the more that we look at them, the more there is a smear between them, and there are big groups of functional types, but then within there's so many subtypes that it might be considered more of a spectrum or a manifold of interneurons rather than separate subtypes that are distinguishable and separable from one another. Read these papers. They're deep, they're long. If you have comments, if you have clues on how to penetrate them, please put them in the comments. Enjoy. And if you liked it, like and subscribe. Thanks for watching. See you later.